Hello, everyone, and welcome to this Sight Loss and Your Genome webinar. It's the third in the series organised by Genomics England in partnership with Retina UK, the Macular Society and the RNIB. My name is Vivian Parry. I'm the Head of Engagement at Genomics England. And if you're thinking, oh, I know that voice, yes, I am also a BBC broadcaster. Now, I'm going to be chatting to Siying Li, a doctor who specialises in ophthalmic genetics, and Gavin Arno, a scientist from the UCL Institute of Ophthalmology. I'll be getting reactions from our panel, and throughout, we hope that you'll be asking questions, which I'll be putting to both our speakers and our panel. We've got an hour together. We are going to finish exactly on time. So let me introduce you, first of all, to our speakers. So let's start with Gavin Arno. Gavin, you're a scientist from the Institute of Ophthalmology. What do you do exactly?
Hi, Vivian. It's great to be here. So my research laboratory at the Institute of Ophthalmology is concerned with investigating the genomics of rare inherited eye disorders. So we analyze lots of the data from the 100,000 Genomes project, and we do new testing and new interesting experiments to identify new and unusual causes of disease for patients and their families.
Dr. Li, how about you? You're at Moorfields, but what are you exactly doing?
Yes, so thanks very much for having me. I currently work as the Ophthalmic Genetics Clinical Fellow at Moorfields Eye Hospital. So I'm a doctor and I see patients with inherited eye diseases in clinic. And I've also been involved with the 100,000 Genomes project for a while, since when I was training in the Southwest. So I was the Genomics champion right at the beginning, where we were trying to introduce and champion genomics within the field of ophthalmology.
Fantastic. Now, we're talking today about long-read sequencing, and lots of you will be thinking, what on earth is that? But before I get Gavin to explain all of that, I want to ask you, Siying, what's the problem that we're trying to solve with long-read sequencing?
So we've had It's an exciting time to be in genomics and ophthalmology, because we've had a lot of advances recently. We've had better technologies, they've become more cost effective, we've found better ways to analyze the data. But a lot of these advances are driven by the development of what you might have heard called next generation sequencing technologies, which are largely a form of short read sequencing technology. And these technologies,
we'll get somebody to, if you're thinking, oh, she's talking stuff I don't know. We're going to explain short read and long read sequencing in a moment.
Yes, definitely. So these technologies are great, and they've really improved our ability to find a diagnosis for patients with rare inherited diseases, but they have their limitations. So at the moment, we might only be able to find a diagnosis in maybe 50 to 60% of all patients, depending on what the condition is. And some of the reasons we can't find the cause is due to the limitations of the current short read sequencing technology. So some of it might be due to, the change in the DNA might be there, but our technology just isn't able to find it. Because some areas of the DNA are extremely difficult to sequence with the current chemistry reactions that our current sequencing technology uses. Some types of changes are very difficult to find with our current technology. So big changes in the DNA, complex rearrangements are difficult to find. And sometimes we can find changes, but we don't know how to interpret them, because we need to know whether these changes are coming from which parents, and our current technology doesn't really give us the ability to find that out. And that's why it's very exciting that we have long-read sequencing technology, which will hopefully be able to address some of these limitations.
So if I can summarize that, we know that the condition that patients have got, for instance, retinitis pigmentosa, but what we don't know is exactly which bit of their DNA has gone awry. And because we don't know that bit, there's lots of stuff we can't do. Like, for instance, we can't start to offer a trial of a new medicine because we absolutely need to know that diagnosis before we can do that.
That's right. And often, if we don't know what the gene is involved, we can't give patients the most accurate information about how their disease is likely to progress. We can't give them good advice about how it's likely to affect other members of the family. So we really try very hard to get the genetic diagnosis so that it can improve the management for patients.
Okay, that's super clear. Now, if you want to ask questions, do put them in the Q&A. Don't put them in the chat, put them in the Q&A, and we'll try and take them as we go along. So, Gavin, this strange thing, long read sequencing, short read sequencing, tell us what that's all about.
Okay, so the human genome then is made up of DNA, and it's 3 billion nucleotides or letters long. And the current technology that Siying was talking about, that's used as part of big studies like the 100,000 Genomes project, is called next generation sequencing. And what that does essentially is smashes that 3 billion letter sequence into tiny fragments, 150 letters long, and then sequences those, reads those, and then we have to try and put all of those back together into one big genome. And that's really difficult in many regions of the genome. So lots of genes, we just can't put them back together because they're too repetitive, or there are lots of copies of very similar genes, which makes it very hard to assemble those. And this is what the jigsaw analogy is about. So if you imagine a very large jigsaw with many tiny pieces, it's
masses of sky.
Exactly, lots of sky, a nice countryside scene with lots of sky. All of those sky pieces look almost identical, and it's really hard to figure out where they go. So what we're able to do now with long read sequencing is essentially smash that genome into much fewer, much larger pieces, so that it's much easier to be able to put it back together and to be able to read through those difficult to read genes. And there are lots of clinically relevant examples of those genes and lots of clinically relevant variants that Siying mentioned again. So things like large rearrangements of big pieces of chromosomes are very difficult to figure out using short read sequencing. So long read sequencing is a new technology that we're able to employ to better understand some of these unusual and rarer types of genetic variation.
So the long read is the equivalent of having, I guess, the toddler jigsaw as opposed to the 1,000-piece jigsaw.
Exactly.
But is the long read always better than the short read, or are both important?
So we think that both are important. Now, one feature of some types of long read sequencing is that the accuracy of the individual letters on those reads is a bit lower than it is for short read sequencing. So I guess in an ideal world, what we really want to be able to have is a combination of the two technologies. We've got the short read sequencing to do our bulk genome sequencing. That gives us the confidence to be able to call variants across the genome. And then when
you say call variants, you mean spot changes?
Exactly, yeah, sorry. And then the long read
I'm just testing him, just testing.
And then the long read sequencing enables us to dig deeper into those bits of the genome that we can't sequence with that technology. But it's a really fast moving field, and as long read sequencing technology develops and the calling of variants, the spotting of variants gets more accurate, we may reach a point at some point in the future where it can, become the primary technology that we can use for genome sequencing. We're not there yet, but that may be. Now,
I know, because you showed it to me earlier, that on your desk you've got a little machine. So show us your little machine.
so this is the little machine that we are using to do all of our experiments in my research lab. So this is a nanopore sequencer, and we're able to use this to replace very large and very expensive pieces of equipment that we may have needed in years gone by. We can now do the same sort of technology with this cool little device. So this is a really, really exciting development for people like me.
And just compare and contrast. I mean, I remember reporting on the first draft of the Human Genome Project, and it had taken about, I think, 10 or 11 years at that point. There were about 10,000 scientists involved. It cost $3 billion for just one genome. So, and they'd use these vast machines, absolutely vast, and huge amounts of computing power. And yet there are you, Gavin, with this little thing, and you can even put it on your phone, can't you?
So yeah, potentially you can plug our MinION sequencer into a phone and be able to process data with that. It's a bit data intense. We do need to use so my laptop that I'm talking to you on today has got some fast processing in it, but it's able to handle all of the data that we can generate with this little machine. But yeah, the first genome sequence, the number of huge machines were in a big warehouse at the Sanger Institute just outside of Cambridge. Now we can replace all of that with this tiny little device. Now we can run complete human genome sequencing on the desk, in an office, anywhere. In fact, there are lots of examples of field laboratories to study pathogen outbreaks in exotic places in the world using this sort of technology in a shipping container laboratory. So really, it's portable, it's flexible, it's a brilliant technology.
So, Serene, what does this mean for you, though, in the clinic? And how are you using this long read?
So at the moment, I think long read sequencing is mainly through a research basis. I think most of our diagnostic testing is through the next generation sequencing technology. But it's very exciting because we do often encounter patients in whom we think we've got the clinical diagnosis, but we can't quite get the genetic diagnosis through our current technologies. And there are some instances that long read sequencing would definitely help. So for instance, in a condition called blue cone monochromacy, it's caused by these very complex changes in a particular region of the X chromosome called the opsin array. And this region is almost impossible to sequence using current short read sequencing technology. So this would be something, these patients would be patients that we might potentially use long read sequencing to get a genetic diagnosis for.
And Gavin, where does it take research? The ability to do this, where does it, what are you using it for? What is it empowering or how is it empowering your research?
Yeah, that's a nice question. So we're doing a few different things at the moment in the lab here, and lots of other people are doing similar experiments. This is not unique to me, but so as Serene mentioned, the opsin array. So that's our kind of model gene that we're testing this technology on, because it is a gene array, so several copies of the same gene in a row. That means that short read sequencing it's a black hole essentially for short read sequencing.
So this is where you're getting repeat after the same little sequence repeated again and again.
Exactly.
And it's like your, lots of pieces of sky in your jigsaw.
Exactly, yes. So we're using now the long read sequencing technology, and we've actually tested a couple of patients in the lab now to see if we can understand what's going on in this gene array in them and in their disease. We're able to assemble that entire region on single long reads. So the short read sequencing is 150 base pairs. We're able to make sequences of molecules. Well, our record in our lab is 250,000 base pairs or letters for a single read. And there's actually this is a bit nerdy, but there's a thing called the long read club on the internet where people are kind of scoring.
I hope you're all taking notes at the moment about this.
So and you know,
long read club.
We can, you know, we can join that club now. We're members. We've got these 250,000 letter long reads that is end to end through this region on the X chromosome. And that enables us then to characterize not only the variants that may be there, but how many copies of the gene are there in individuals, what sort of rearrangements there are between those genes, because that's another common mechanism of how these genes are disrupted as well. So this is really quite new stuff. This is haplotyping, and sorry, that's a very technical term, but being able to read through the individual copy of the X chromosome is a really quite a new thing to be able to do. So we're pushing.
And the point is here, isn't it, that Gavin, that sometimes it's the number of repeats that are important in not only the diagnosis, but also perhaps the severity of a condition. Is that right? Is that holds true for eye disease as it holds true for other things? For instance, some of the muscular dystrophies, I think, are.
Yeah, so slightly different to I think what you're referring to is repeat expansion diseases there. So slightly different to that in the opsin array, where we can have a variable number of the opsin genes in that array. Only the first two are made into proteins, and these are, so the opsins are actually the proteins that capture photons of light in the retina, and each cone expresses a different color opsin. And it's the red and green opsins on the X chromosome. And so the first and the second genes are made into proteins, but you can have more copies of them downstream of that, so further along in the array. So what's really difficult to be able to understand, if we find a variant, is being able to figure which copy of the gene that variant is in, because it's got to be in the first one or two genes to have an effect on the actual expression of the gene. So that's something that's impossible with other technologies, and long read sequencing is really opening the door to our ability to characterize these things. And you know, these are early experiments, so that's one example of something that we're doing in the lab now.
So how excited does it make you, Siying, when you hear Gavin talk about his long read? I mean, he's away with his long read club. Is that, you know,
Yeah.
Other people have the Thursday murder club like Richard Osman, but he has his long read club.
Oh, it's extremely exciting news. And I think we're very lucky to be working in a field where there are these exciting developments happening all the time. And I think, you know, it's not just the opsins that Gavin was talking about as well. There are other areas in which long read sequencing can really bring benefits to patients as well. So another common example that we encounter is where we find changes in the gene, but we just don't know how to interpret them accurately. So for instance, in certain conditions known as autosomal recessive conditions, you need both copies of the gene to be not working so well. So both copies need to have changes. And we all have two copies of every gene in our body. One comes from mom, one comes from dad. And in autosomal recessive conditions, both copies need to be not functioning before you develop the condition. So sometimes we find two changes in the gene, but we just don't know if these changes are occurring one on each copy, or if you have two changes on one copy and your other copy is working fine. And so it's very frustrating in these instances because we think we've got the answer, we just don't quite have enough evidence to show it. And then it's very difficult to explain to patients as well that we think we've got the answer, but we just don't have the evidence to say this is your genetic diagnosis. And again, this is another field that long read sequencing will really be able to help. So it's very exciting to think about the possibility to get to achieve a genetic diagnosis for all these patients who we previously couldn't.
Now, I mentioned before that this is kind of the first stage, actually finding a diagnosis for somebody.
Yes.
And
yes.
what everybody listening will want to know is, okay,
Yeah.
I've now got a genetic diagnosis. That's helpful to my family. And you now know exactly what's causing my condition. But what next? What does it mean for developing treatments, for instance? And I should say, I think at this point that, I mean, from my point of view, I often report on things where visual loss seems to be at the back of the queue in terms of research funding, but actually you're at the front of the queue, and you're right at the cutting edge in terms of new therapies based on genomics, because the eye is just such a special organ that you're able to do things that other organ systems just can't possibly achieve. So what are you thinking about next once you have these kind of diagnoses?
That's right. So in fact, there is one gene therapy that is licensed for an inherited eye condition. This is for a specific condition caused by changes in the gene known as RPE65. So we have one therapy already, and we've got other therapies, other similar gene therapy treatments that are in clinical trials. But often these are gene-directed therapies. So you need to know the exact gene involved, and sometimes you even need to know the specific spelling change in the gene to be eligible for these trials. So that's important to get the genetic diagnosis for these patients so that they would be eligible for the trials. And then in the broader concept, if we understand a little bit more about how spelling changes in the gene can affect the function of the gene, we can then learn a little bit more about how disease pathways, how the mechanisms of disease develop. And a better understanding can lead us to develop ways of targeting it and to develop new treatments in the future. And I know we're talking about inherited eye diseases, which are rare conditions, but they involve degeneration of cells in the eye. And there are lots of other processes where you get degeneration of cells in the eye. So the longer term hope is that if we understand these disease pathways through studying rare diseases, we can then understand how to target these pathways and hopefully treat many conditions which affect the eye in future.
So there you are, people with rare eye disease who are listening to this webinar. You are absolutely trailblazers.
Yes.
So Gavin, apart from being ambitious to do 300,000 base pairs or 500,000 so that you will get, super membership of your long read club, what's your next ambition? What would you like to do? I mean, Serena's already mentioned this idea that actually what you're doing is also helping with the understanding of the mechanisms of the eye. But what else would you like to achieve?
Yeah, just to expand on that a little bit. So another experiment that we're doing in the lab now with long read sequencing. So long read sequencing is also known as single molecule sequencing. So not all reads have to be long, but what it enables us to do is to see sequence at the molecular level. So we're able to see individual molecules of DNA and to read those, which compared to traditional technologies which rely on generating a big signal from lots and lots of molecules of DNA. And so one thing that has enabled us to do is to take what we call variants of uncertain significance. These are some of the patients and participants of the 100,000 Genomes project may be familiar with this term. It's a variant that's been identified in a gene, but we don't understand what it's doing to the protein.
So we think it's suspicious, but we're not really certain enough to say to somebody, this is the actual cause.
Exactly, yeah, and this is a big problem for molecular genetics. This is so lots of variants are put into this category because we don't have enough evidence, we don't have enough understanding of how they might be affecting the protein or how they might be causing disruption. And so we're now developing assays to be able to test those using long read or single molecule sequencing in the lab, which we can feed back into the clinical laboratory. And what's key for eye disease genetics is that many of these genes are only made in the eye. So they're only made into proteins in the eye because that's the only part of the body where they're needed. And that means that typically we would take a blood sample to be able to look at these genes, but it's quite hard to do that when we're trying to interpret variants of uncertain significance from the blood because the gene just isn't expressed there.
So just to make sure that I've understood you, so it's a bit like the genome is this huge big thing, and you could think of it like I suppose a recipe book or something. So in some cells you only ever get the spaghetti bolognese recipe.
Exactly, yes.
So it's no good when you look in a blood cell to look for the bolognese recipe because that's only in the eye cells.
Exactly, yes. And so we're able to break that rule a bit. We're able to cheat and look at the recipe book in a different way with single molecule sequencing now, and to be able to understand what's happening there. And as we learn a bit more about how that method can work, how that assay can work, we can feed that back into the clinical lab, and we can help the clinical lab be able to interpret these types of variants and get more answers to more patients. And this particular type of variant, we call them splice variants. These are also quite amenable to genetic therapies. So there's a type of therapy called an antisense oligonucleotide, and what that is it's a tiny little piece of, it's RNA, it's not actually DNA, but that will stick to the affected part of the gene and stop it from having a bad effect on the gene. And so non-coding or splicing variants are amenable to that type of therapy, and there are lots of studies looking at how these can be developed. So we're better understanding how variants affect genes and how variants lead to disease, but also the more that we understand about this type of variant and the more that we identify, then we will grow in confidence as to how we will be able to develop therapeutics for those types of variants in the future as well. So I think the future is quite bright, and that is exciting for me, definitely, as a scientist.
So you're looking for the needle in the haystack, except you find lots of needles in the haystack. Then you've got to work out which of those many needles is the needle in the haystack that you're really looking for. But the real prize here, from what I understand, is that if you then find it, then there are potentially these ASO, these antisense oligonucleotides. I hope you're writing that down, folks, because it's a very long word, but ASOs, and there are more and more of those coming along. And there's a real possibility that if you found those particular changes, you know they're definitely the ones causing the problem, then you can develop these ASOs just to sort them out. I mean, it's extraordinary technology. It's really extraordinary technology, and it's long read that it's enabling you to do that.
In many cases, that's true. I mean, there are examples that we already understand, and these have been figured out from various other technologies. But I think the key with long read sequencing is that it's accelerating these sorts of discoveries for scientists like me now.
And making your life a lot easier, Serene, because you'll be able to help more patients.
Exactly.
Fantastic. So let's bring into the discussion, if we can now, two people who are really important. The first is Dave McCormick. Dave, welcome.
Thank you, Vivian. Well, thank you very much for inviting me.
Dave is a member of the Genomics England participant panel and hugely enthusiastic and important member of that panel. And also we have Matt Carr. Now, Matt is the, have I got this right, the Information and Support Manager for Retina UK. Matt, you're very welcome, and thank you to Retina UK for your support for this event. It's really appreciated. So, Matt, let's start with you. What did you make of all of that?
It's so exciting. I also live with inherited sight loss myself as well. So not only from our community, but from a personal perspective, knowing that where we've had people with no genetic diagnosis in the past who maybe haven't been eligible to look at clinical trials and such like, this just opens up so many different avenues for people going forwards with their own sight loss journeys.
And it's not as, I mean, I know people are put off by, you know, long read sequencing. It sounds super complicated, but actually it's not so very complicated when you get down to it. Dave, what did you take from all of that?
Thanks, Gillian. And like Matthew, I agree. I think this is really very exciting. I myself also have a lived experience of an inherited sight condition. And I think for me, for the first time in quite some time, I've been involved as a member of the participant panel and a participant of the 100,000 Genomes project, that I'm actually starting to see an end point, or a point at which all this amazing work that's been going on over the last 6-7 years is going to evolve into some kind of meaningful, tangible outcome. Because for me, the technology is great, but as a patient, what I want to know is, how is this going to help either me, my family, or other people with my particular flavour of sight loss, microanophthalmia? How is it going to help me and others like me coming up through the ranks in the future to improve quality of life, to improve diagnosis, to improve how we can either stop deterioration in vision and hopefully, excitingly in the future, return some vision that's lost?
Serene, what do you make of that? Because I mean, there are lots and lots of these conditions, but some of them are presumably more amenable to this kind of, you know, gene therapies. And are we talking about restoring vision, or are we talking about correcting the problem altogether? What's the options?
I think at the moment, a lot of our efforts are based on stopping things from getting worse. So there are certain conditions that are more amenable to those. So conditions where your retina still maintains some structure and some of the cells are still present so that they can receive the gene therapy treatment, which is often where you're trying to replace, add more functioning protein where there wasn't before. And then you have certain types of mutations, like what Gavin was talking about, that are more amenable to this new therapy, the antisense oligonucleotides. So being able to understand the genetic basis of a condition will help to determine which patients are eligible for the current type of treatments that we can offer. But I think even so my understanding of structural eye disease, including anophthalmia and microphthalmia, is that quite a proportion of them are caused by structural variations in the DNA. So these are large changes in the DNA.
So missing bits, or
additional bits, or bits that have been chopped up and switched around and put back in. And these changes can be more difficult to find with our current sequencing technologies, which are a bit better at picking up sort of single letter changes in the DNA. So in the first instance, the long read sequencing technology at least can try to help us find the diagnosis and to find, and also as we get better with our technology, we can look for new genes that can cause eye disease. And then a better understanding of new genes will allow us to better understand additional pathways that are involved in, say, the development of the eye or the development of the eye not working so well. And all these will lead to treatments in the future. So I think that we have had some advances. We're still in early stages trying to treat all forms of eye disease, but or all forms of inherited eye conditions. But there are definitely lots of clinicians and scientists working very hard to take us to the next step.
Now, some eye conditions are inherited eye conditions are evident almost from birth, but some are not. And do you think that in the future, whole genome sequencing can be used in newborn screening? I mean, it's something that Genomics England is involved in. We don't yet know what conditions are going to be included, but do you think that's a future possibility, either Gavin or Siying, to include some eye conditions? Because perhaps if you gave these treatments earlier, you might salvage sight loss before it occurred.
I think
do you want me to answer that?
that's quite a controversial issue. I think there's lots of discussions
It is. I
for
mean,
both sides of the
technology
argument.
The
Yeah,
technology is certainly there.
yeah,
The whole genome sequencing, to be able to do that, it's a bigger question than that, though, I think. But just before we move on, though, I just wanted to add to Dave's point that it's important to know that long read sequencing is not all we're doing. It's just one string to our bow. There are lots and lots of researchers and clinicians working on the 100,000 genomes project data all the time. So even though you may have got a negative report back from the first step of the analysis, that data is being reanalyzed by lots of researchers and scientists and clinicians all over the country to feed back and update reports for patients and participants as well. So long read sequencing helps, but we're also doing lots of reanalysis on the short read data as well.
yeah.
And Dave, you're absolutely on the money there, aren't you? I mean, you're pursuing diagnosis for people. There's a lot of pressure from the participant panel to make sure that as much sequencing, you know, the search goes on. So although the project has finished recruiting, it's certainly not finished.
Absolutely, Gillian. It never ends. The genomes may have been collected, 100,000 plus genomes, but now that we've got these samples, which you know, like myself, people have donated that. It's what we do with those samples, and the technology, as it's evolving and changing all the time, and the techniques for reading the results from that technology is also changing and evolving all the time. So it's not a static picture. And as Gavin's quite rightly pointed out, you know, you might get a negative or a no response at this point, but that does not mean to say that we won't get, or the individual patient may not get, a positive response in the future. It's only as the tech improves and the way we read the responses improves, that picture may change. And it's hugely reassuring and very encouraging to see these changes evolving all the time, moving from rooms of computer banks to a device which can fit in the palm of your hand, and costs coming down all the time. It will be only a matter of time before I think solutions will be found with the use of this long read tech.
Matt, do you have this sense of eye conditions actually being in the front of the queue for a change, instead of behind the door, with, you know, not given so much research funding, but now a lot of these things are really crystallizing in the visual loss community first of all?
I think so. I mean, there's a huge amount of work with various different groups looking at kind of trying to prioritize various areas of genetic medicines. One thing that's always frustrated a number of people in the sight loss sector is, particularly around research, is that they spend more money on why you lose your sense of smell than you do on any kind of sense of any of the eye conditions. So I think, as I said before, it's very, very exciting, and I do think there is a lot more emphasis being placed on some of these genetic eye conditions, particularly off the back of the data gathered from the 100,000 genome project.
Now we've got a question here. I don't know who wants to answer this. It's from Neil Ward. How many people with vision disorders are included in the Genomics England project? So it's being added to all the time because the Genomic Medicine Service now is up and running. But how many came in through the 100,000? Siying, do you know the answer to that?
I'm not aware. I think it's one of the more common categories, but I don't know the exact number. Gavin knows. Okay, hand over to you.
So ophthalmology represented 8% of the overall patient recruitment, and I think there were about 3,000 individuals with sight loss in the 100,000 Genomes project.
And actually, I think we should do a shout out here. If you, I mean, a shout out to the charities and to all their supporters, is that first of all, if you're part of the 100,000, thank you, because your data is so valuable. But second, if you know of people who are asked if they would consent to whole genome sequencing, do please say to them that there's a second question, which is, would you like to take part in research? And we'd like so many more people to say yes to that, because although eye disorders, there's relatively speaking a lot of eye disorders in the data set. Actually, because some of these things are very rare, it means you've only got small numbers of some conditions. So we'd really like people to agree to put their genomes in research. And are people usually very sympathetic to that, Siying, when you ask them?
Yes, I
how
believe.
Could anybody refuse you? But
No, so our patients are very altruistic, I think, and they often understand that when they participate in research, the findings are still some years away and they might not directly benefit our patients, but they're very keen that we gain knowledge so that we can find out more about inherited eye conditions for future generations. And I would again put a plug out for consenting to having your data stored in our research library, because we are learning more about genetic eye conditions and how changes affect the condition all the time. And say with long read sequencing, we're finding new, gaining new understanding that we can then apply to all the data that we've got from our previous short read sequencing. And we do reanalyze the data. We try very hard to keep looking. You know, the journey doesn't stop once you've had the testing done, and there's no initial finding. We do keep trying to reanalyze it. So if your data's in there, you know, it might be some years down the line, but hopefully we'll be able to find the diagnosis in future. And also, as you mentioned, there is power in additional individuals with rare conditions being in there. So we can look at a sample of patients with the same change in the gene, and we can see if there are certain patterns that develop. And this will then help us to provide advice for all the patients with that same gene. So it really does help us a lot.
And Gavin, how important is the data to you in your work?
It's phenomenally important. So the 100,000 Genomes project is a global leading project. So at the time it was launched, it was hugely ambitious. It was the first study of its kind in the world to look at this sort of number of genomes from rare disease and rare cancer families. So for a scientist like me, a molecular geneticist, sorry, I don't even know my own speciality, it's it really is game changing. So, you know, being able to analyze and access all of this data in the ways that we're able to with working with Genomics England, it's revolutionized the way that we can work as geneticists. So, you know, I can't emphasize enough how important this study is for scientists like me. And just to echo the thanks to all the participants and all the people who consent to include their genome data in the research embassy. this is the only way that we can make these big discoveries now, is to be able to work on large data sets and lots of individuals in one study. So yes, it's very, very exciting, and it's changed the way that we can do the sort of science that I do.
I mean, that's a fabulous thing to hear, isn't it, Dave?
It's very reassuring that, you know, we need to keep encouraging people to contribute the genome sample with all correct and due security and protections with those samples. And who knows what's just around the corner. For me, one of the big exciting things I'm hoping to see is that with this long read technology, it will move from the centre out to the different regions, to the different genomic medicine services, so that, you know, wherever you live in England, people will be able to get access to that technology. And apologies to colleagues from Wales, Northern Ireland and Scotland who've tuned in, but the genomic medicine services across England, I'm hoping that this tech will evolve from the centre outwards, and so that everybody eventually will be able to get access wherever they live.
And people sometimes ask us, why do we need to collect data about people's health, and why do we go on collecting that data? Isn't the genome sequence enough in itself? But actually, the thing about genomics is that you need to know what happens next. So did your site continue along a kind of a plateau, or did it suddenly get worse? And actually those differences can be really important, can't they, Serene, in helping decide exactly what conditions people have, and also where to look in the genome.
Yes, that's right. So even if we know the gene affected, we're starting to learn that different changes can affect the severity of your eye condition. So if we can study lots of people with different types of changes, we can then gain a better understanding of whether or not these changes are likely to cause more mild disease or more severe disease. And also, some genes we're learning don't just affect the eye, they can also affect other systems in the body. So if we can collect the health data, and then we go back and look through it, we can start looking for patterns in terms of which other systems might be affected. And this has important health implications for anyone who we find a change in that particular gene, because it might be that they might need to be offered screening for particular health conditions. If we then learn that, say, if your heart is going to be, if a small percentage of patients with this gene that causes an eye disease also develop heart problems, then we know that these might be patients who might need to be referred to a cardiologist for a follow-up. So yes, it's important to collect health data, and then for us to tie it in with the genetic data.
And that's an example really of where a genetic diagnosis can help us with prevention, because it may be too late to retrieve sight, but we can certainly prevent people from having heart attacks as they get older. We know how to do that bit.
Yeah, or it can impact other members in the family as well. So you might find a condition in one person, but then if we know that other members of the family are at risk of being affected, we can then screen for the gene in them and to address all these other issues before they develop.
So we're coming towards the end now, but if anyone has got another question, then do please put one in. We've got a technical question here, which I think is for Gavin, and it's about AI and machine learning. So AI, I mean, ChatGPT absolutely in the news, but of course, machine learning is really important in the analysis of genome data because there's just so much of it. And this question is, do you always remove the data that's low quality, or can you use some computational method or AI to not lose the data that's low quality? That is a very tech question. So let's have a short answer.
So we remove data that's very bad, and we're developing our assays so that we generate enough data so we can trim off the poor quality stuff and only need to look at the good quality stuff. I guess that's the simplest answer I can give for that.
Because it's not always easy to read. I mean, we're thinking it almost like a book, you know, everybody's book arrives, we just open the book and start, but it's not like that.
But also, you know, the technology is getting better as well. So the past couple of years, the quality of read quality from nanopore sequencing data has improved hugely, and there are new developments in the pipeline. So we haven't tested out some of the newest versions of the chemistry yet, but there are lots of new improvements, and the data is just getting better and better. And there are other companies as well making similar technologies as well, whose data can be incorporated with nanopore sequencing. And this is something that big global whole genome. So there's a project called the Human Pangenome Project, which is combining short read data with different types of long read data to really get the gold standard of genome sequencing from that as well. So that's something that's developing and ongoing as well.
So our question has come back. So you're creating more than you need, effectively.
Yeah, I mean, we're still learning how much we need really with nanopore sequencing data. It's a very new technology. It's not in the clinical realm yet. So this is all research data that we're generating, and so the strength of the data is that we can generate plenty of it, so that we are able to have enough confidence to be able to make these calls in the research lab, and we'll feed all of this back into the clinical lab, and then the next step will be around validating the technology as a clinical tool and so on. But that's a little way off yet.
Where do you think, I mean, this is the question that is always the $64,000 question. Where do you see long read taking us, say, in 5 years' time? What do you think we will look back in 5 years' time and say long read technology has given us? I'm looking at you, Gavin.
So I was at a meeting recently talking about the use of long read sequencing to understand the whole human genome, because currently, so we talk about whole genome sequencing as if we are actually sequencing the whole genome through the 100,000 Genomes project, but we're not. We're missing huge chunks of it out, and that's important for scientists like me, and also is important for the clinic. So I think, you know, long read sequencing in its various forms will give us that information. So the first genome was finished in 2001, and now we're on the cusp of finishing the first real genome, actually, with projects like the telomere to telomere project and the Human Pangenome Project. They've only got one haploid genome so far with those studies, so that's 1/2 of a genome. So we haven't even got a full genome yet. So that's what long read sequencing will do.
So have I been fibbing all this time?
Absolutely.
Saying that the whole genome has been sequenced.
So we talk about whole genome sequencing, Illumina short read sequencing being able to cover roughly 95%. But the truth is, you know, we don't really know how much we're missing with that technology because it just isn't there. So that's what long read sequencing will certainly give us. It will give us more confidence in variant discovery and molecular diagnoses within the next 5 years, I think. It will, you know, there is an argument that it may eventually replace short read sequencing, as I think I mentioned earlier. So that's probably a bit further away than 5 years, but that could be the longer term future, is that it is a clinical tool that's readily available on the NHS.
So Gavin is very excited about the research side, Siying. What about you in the clinic? In 5 years' time, what do you think long read may have achieved for you in the clinic and for patients?
Well, I think we're already seeing examples where Gavin's work with the long read sequencing has given us the genetic diagnosis in patients that we couldn't previously achieve. And these are just limited numbers at the moment, but hopefully we'll be able to expand that and to be able to provide a genetic diagnosis for more patients. And then, as Gavin mentioned, we've got a lot of research ongoing into splicing changes that are amenable to different types of therapies, the antisense oligonucleotides. So hopefully with long read sequencing, the more it becomes available, the more splicing defects we'll be able to study and understand, and the more we can identify, and this will expand our pool of patients who would be eligible for such therapies if they are then shown to be of benefit. So very exciting times.
And Matt, how interested are your members in this kind of genomics?
Really, really interested. I've actually got a question. I wonder if I can just slip in. I know we're not looking at individual cases. I think, Gavin, you've answered half of this question here. So this is something we hear a lot of. You know, I was part of the 100 Genome Project and I've not had a diagnosis. What happens next? So I think, as you said earlier, that data is constantly being rerun. So obviously there's hope for diagnoses in the future. But this one is around someone's gone for genetic counselling and their son has a few small black dots on their ERG, their one of their tests in the clinic. So what the clinician is trying to do is to gain access to the data from the 100,000 Genome Project for this particular individual. How can they do that, and is that possible? And is there anything else that that can be done to maybe speed up and make this process a little bit more efficient?
Yeah, that's a really good question, and it's a question that we do face, and you know, clinicians and colleagues and scientists here are accessing that data and trying to link it up with the patients that they see in clinic and be able to interpret that data for their own patients. So it is possible that clinicians can access the data. So the guys at Genomics England who are on the call will be able to correct me if I'm going too far off course here, but they need to register as users of the research embassy to be able to access the data in the research embassy, and that involves submitting a project proposal and having an institutional agreement with the Genomics England. And so once that's in place, then you can log into the research embassy and you can analyze all of the data in any way that you want. So the data is anonymized, so you can't link from the research embassy to individual patients directly. So that's important to note.
Although the NHS can do that if they want to look at one particular person.
Yes, so researchers can't do that, but the NHS can. So if a clinician has recruited a patient to the study and they're keen to understand what the data means for their individual patients, then via the NHS link with the data, they're able to look at that case directly and interrogate that data directly. So yeah, there are ways to do that.
A very quick last question. How does the pharmaceutical industry come in contact with the research results to specifically create more personalized treatments, genetic diagnosis? And perhaps I can answer that one by saying that we work a great deal with different both biotechs and bigger pharmaceutical companies who have interests in different conditions. And if they have something that they think might be appropriate for a particular disorder, then they contact us. They ask us to find the people that have that particular alteration that they're interested in. And then we at Genomics England write to the clinicians of those people and say, this company is interested in a trial. Is your patient appropriate, suitable? Because the clinician will know, whether their disorder is suitable for trial, whether other health problems mean that they can't be on it, all those kind of things. So we work very closely, and I think lots of people are anxious about pharmaceutical involvement, but actually it's a very important way, because it's obviously the pharmaceutical companies that develop the new drugs, not the NHS. But yes, we're closely aligned with them. We're now going to finish. I'm always going to leave the last words to Dave McCormick. And perhaps you could thank our panelists and on our behalf.
Most certainly. Thank you, Vivian. Firstly, can I pass on my huge thanks to Dr. Gavin Arno and also to Dr. Siying Li. Thank you so much, both of you, for your amazing insights, your technical, being able to explain the technical language into plain English. Very grateful to you. Can I also thank Matthew Carr? Matthew, thank you to you, to the Macular Society, and also to our friends in the RNIB and, sorry, your Retina UK and the Macular Society, I'm so sorry, and the RNIB. And also, not least, to Viv, our amazing chair today, and also to Dr. Charles Stewart and Zan Sargent and the Genomics England comms team. Thank you, all of you, for an amazing event. I think it's worked really well. I hope the listeners out there, the viewers, you've enjoyed it also. You've managed to get some benefit from the discussion today and the previous events. And I'm very proud to say that as a result of this model, we hope to go on and use this approach in supporting other people in other areas of GSIP research. So bear in mind, you were the first. We hope to use the success going forward. Thank you all very much.
Thank you.
Visual loss at the front, not at the back. It's very good to know. And to all of those of you who've listened to this, thank you so much. Goodbye.
Thank you all.
Thank you.